you and I had spoken, offline about touching on a technology called CRISPR genome editing. And if you could spend a few moments for us and just explain what that is and why it's good or not good.
So CRISPR is an anagram for basically a process that is best described as gene editing, which means that you can go into the DNA and make changes, make edits. And in adults, that's sort of less, if you change the DNA in, say a few cells, that's may or may not have a great impact. But if you want to eradicate, say, genetic diseases, it's possible to use this technology. If you have, say, Huntington's disease in the family, you could potentially go into an egg from, or a sperm from one of the parents or whoever carries the disease and correct the mutation. So you can edit out the mistake. So that's the gene editing piece. It's not a destructive editing, it's a correction of these genetic diseases. And
you do that like once it's discovered, or in anticipation of it, because you.
So once you know what the mutation is in the family, and then the family wants to have more children, you can go into the egg or sperm of the couple and correct the mutation. You can go and find the person who's carrying the mutation, go in there and correct it so that the child would not be affected. Otherwise, half the children would have the disease.
Right.
But it's possible to go in and avoids all the issues of abortion and that sort of way of managing the problem. But you can go in and actually correct the mutation.
And is that happening right now?
Yeah. So it is. There are a few reports in China where it's been done in humans. There's the issue that's come up is that this tool that's when you focus it at this mistake to correct it may fix that, may make that correction that you're interested in, but may induce some mistakes elsewhere just because it's hard to control and focus it. So it's important that once you use the tool, that you then go back and sequence the entire genome of the cell that you've edited to be sure you haven't made any other mistakes. And we were only now appreciating that, and it's not clear whether these experiments in humans that were done in China were adequately managed or sort of sequenced to see what sort of mistakes they had introduced.
Or maybe caused other mistakes.
Sorry?
Or maybe caused other mistakes.
Exactly, yes, right.
And along the same genome concept, how does the CRISPR part fit into that?
So that's the tool. That's the actual sort of enzyme and DNA sort of specificity that allows you to do the editing. CRISPR is really just it's the type of enzyme that you use to do the editing. It's an anagram for some sort of palindrome sequence that's especially useful for editing.
And when someone says, "Hey, I had my genome mapped," mapping genomes, the services, the companies that do that now, right? Why would you want that done? And actually, have you had that done?
I haven't done it well, actually, I guess I have. I've had the 23andMe thing. I've used that for patients where I've been trying to find a cause of their illness, and it's now possible again because of the big data to have a database that contains every DNA mistake that's ever been associated with cognitive impairment in any disease ever since DNA sequencing was available. So if you have someone with cognitive impairment and you can't figure out what it is, you can take that person's genome sequence it completely and compare it against this database and see if there's anything there that gives you any clues as to where, what genes might be defective and how, and that might then give you point you towards the cause.
Interesting, and as these developments continue to, take form and accelerate, I would, and that's, part of what I want to communicate, I would assume, and you would already know this, that people with more financial resources might have quicker access to these technologies than folks that don't. Is that like the case right now? And you kind of see that being versus some of these cool things being like mainstream?
I mean, certainly in terms of both access to care and to the best care, yes, that's certainly, there's a socioeconomic stratification for that. In terms of being potentially targets for CRISPR correction, maybe not necessarily. I mean, for example, sickle cell is one disease that's being thought of as a disease that could be cured by CRISPR.